Hello everyone, welcome back for our last topic for this semester, sex-linked traits, coming out of section 12.2. This is the only section in chapter 12 that we're going to be covering. All right, so in humans and other mammals, biological sex is determined by the sex chromosomes, and an individual under normal circumstances is going to have two sex chromosomes. The XX combination is biologically female. The XY combination is biologically male. Now, there are some other relatively rare sex chromosome combinations that result from incorrect chromosome separation during meiosis, so that a gamete may actually have two sex chromosomes instead of one. That can potentially result in the XYY combination or the XXY or the XXX combination. And so individuals with those relatively rare combinations are going to have some developmental abnormalities and just some different physiology that they're going to have to deal with. It is worth noting that the XX and XY sex chromosome combinations are specifically for mammals. Other animals, biological sex is determined differently. So in birds, for example, it's the female, not the male, that determines the biological sex of the offspring. And bird chromosomes are referred to as Z's and Y's. So just be aware that this XXXY combination is not universal throughout the animal kingdom. Okay, so for the human sex chromosomes, when we look at this scanning electron microscope, we see that the X and the Y chromosome are very different from each other in size and in shape. Okay, they also carry vastly different amounts of information. On the X chromosome in particular, there are a lot of genes on the X chromosome that don't just deal with biological sex. Okay, and we're going to talk about those a little bit more later. So this figure, again, is just reminding us that mammalian sex determination is determined by the male, because biological females, their only sex chromosomes are XX. And so when females make eggs, the only possibility for the eggs is to have an X chromosome. Whereas men are XY, so their sperm cells are going to have the 22 autosomes plus either an X or a Y. Okay, and so depending on which sperm fertilizes an egg, the offspring is either going to be XX or XY, assuming everything went properly in meiosis and there are no other genetic abnormalities going on. All right, well, this is a map of the human X chromosome, and you can see that there are lots of genes on this chromosome. And many of these genes are known to cause diseases if there are mutations, specific mutations in these genes, okay? And so sex-linked traits, or X-linked traits, to be more precise in our language, occur more often in men because biological males only have one X chromosome. And so if the one X chromosome that they have has a mutated version of an allele that causes one of these X-linked disorders, biological males do not have another X chromosome to compensate for it, okay? So when it comes to genotype and phenotype, males cannot be heterozygous for the genes that are found on the X chromosome. They are either going to have the dominant allele or the recessive allele for traits that are inherited in that Mendelian fashion. Okay? Biological females, on the other hand, can be homozygous dominant for the dominant sex-linked allele. They can be homozygous recessive. They could have 2 copies of the recessive allele for a particular X-linked trait, or they could be heterozygous. Okay? So biological females have more genetic possibilities because they have those 2 X chromosomes. Now, when we're talking about transmission of X-linked traits, this example is just dealing with colorblindness as an example. Okay, and let me just kind of walk you through how the terminology is used here. Okay, when we're dealing with sex-linked traits and we're doing Punnett squares and things, we have to use our X's and our Y's because these traits are carried on the X chromosome. They are sex-linked. So we can't just use the first letter of the trait, like a C for colorblindness, because we have to show that that trait is carried on the sex chromosomes. All right, so the way that this textbook is using the terminology, a woman that has normal vision would be written as X big N, X big N, okay? Normal vision. A man who is colorblind would have X little n, Y. And notice that the allele letters are superscripted, okay? They're higher than the regular X's and the Y's, just to help you keep track of it better, okay? So if you have a woman who is homozygous dominant for normal vision, and she has a child with a man who is colorblind, all of their children will have normal vision because the mom, the only X or the only colorblind or the only color vision trait that she can pass on is the dominant allele. So all of their offspring will have the dominant allele. Now, their daughters, if they have any daughters, will be heterozygous. Females who are heterozygous for X-linked traits are referred to as carriers because they can pass on the trait, but they don't exhibit the phenotype, okay? If we look at a cross in the next panel with a carrier woman and a man with normal vision, and we do the Punnett square, we're going to see that because the man has normal vision, all of their daughters will have normal vision. Because what does a male donate to his daughter? Only his X chromosome when it comes to sex chromosomes, okay? So all of their daughters, right off the bat, we know are going to have at least one copy of the normal vision gene, okay? Now, some of the daughters, 50, there's a typo, sorry, there was a typo in this figure, because this daughter right here, that should be a capital N. Half of the daughters will be carriers, half of the daughters will be homozygous dominant, okay? But what about the sons? Because mom is a carrier, if an egg that has the recessive allele is fertilized by a sperm carrying the Y allele, then that son will be colorblind, okay? So women are said to be carriers of X-linked traits. X-linked traits actually show up, the phenotype shows up more often in men because of this situation right here. Because a son gets from his father the Y chromosome, The Y chromosome has nothing to do with X-linked traits. The son only gets one X chromosome from its mother. If the mother has the recessive allele that is passed on in the egg that is fertilized to produce that son, then that son is going to have the phenotype. Okay? Well, what about if you have a carrier mom and a dad who expresses the phenotype? In this case, if they have a daughter, they would expect a 50-50 chance that the daughter would be colorblind. Okay? Same with the son. Okay? So we would expect that 50% of their children would be colorblind. Okay, now we're going to practice a couple of Punnett squares just to make sure that we understand how to do Punnett squares for sex-linked traits. So let's talk about a different sex-linked trait now. Let's talk about hemophilia. Hemophilia is an X-linked trait dealing with the inability of blood to clot properly. So let's have a carrier mom who has children with a non-hemophiliac male. Okay, so again, the capital H is the normal blood clotting allele, no disease. Lowercase h is when that is the recessive allele. And if somebody has just the recessive alleles, they are going to exhibit the phenotype for hemophilia. Okay, so remember when we're setting up Punnett squares, we put the gamete possibilities for one parent across the top and the other parent down the side. Okay, so let's put mom down the side. So mom, in her egg cells, she can donate either an X with a big H or an X with a little h. Dad, half of his sperm are going to have X with the big H and half of his sperm are going to have a Y. Okay, well, this top left square, this potential offspring would be female. She would be homozygous dominant, completely normal when it comes to blood clotting. The bottom left offspring, we would predict to be biologically female. She would be a carrier of the hemophiliac gene, but would not express the phenotype. All right, top right box, biologically male, XY. Male would be normal from a phenotype perspective because he inherited the X chromosome with the dominant allele, no disease, no hemophiliac disease associated with that allele. Sorry for the barking dogs. And then the bottom son, bottom right, would be a hemophiliac. He would express the phenotype for the hemophiliac disease because he has, from his mother, the X chromosome with the recessive allele. Okay, so he would express that phenotype. Okay. Sorry, dogs were distracting me in the background. All right. Well, when we're talking about Punnett squares for human offspring and such, we've got it on your exam. I may ask you a question that might be, what percentage of this couple's children would we expect to have hemophilia? Okay, well, if the question specifically stated children, then we would expect 25% of their children to have hemophilia. Okay, because they have a 1 in 4 chance every time they have a baby. They have a 1 in 4 chance of having a hemophiliac baby. Okay, but if I ask the question, what percentage of the couple's daughters would we expect the hemophiliacs? I'm so sorry. The answer would be 0, because none of the daughters are going to express the hemophilia phenotype. Okay? If we ask the question, what percentage of the couple's sons would we expect to have hemophilia? The answer would be 50% of the boys, right? Because every time you have a baby, you have a 50% chance whether it's going to be male or female-ish. But if we're just looking at the boys, if they have a son, he has a 50-50 chance of whether or not he's going to have hemophilia, okay? Now, we can combine Punnett squares for sex-linked traits and autosomal traits, okay? Autosomal traits are traits that are carried on the autosomes. And remember, the autosomes are all the other chromosomes that aren't sex-linked chromosomes. Okay, so let's just play pretend that we have I'm having technical difficulties this morning, that we have a mom who is a carrier for color blindness, and she also happens to be able to roll her tongue. Okay, and let's just say she is heterozygous for tongue rolling. And she's going to have a baby with a man who is colorblind and cannot roll his tongue. Okay, so how are we going to set up the Punnett square? Remember, when we're doing a dihybrid cross, we're going to have a bigger Punnett square, and it's going to have to be a 4x4 square. Okay, so let's put Dad's gamete possibilities across the top. Okay, so Dad could have a sperm cell that has the X chromosome with one of the little r's, or the X chromosome with the other little r. Or his sperm could have the Y chromosome with one of the little r's, or the Y chromosome with the other little r. Okay, well, Mom over here, her egg cells could have the X chromosome with the normal vision allele with the dominant tongue rolling allele, or, the X chromosome with the normal vision allele with the recessive tongue rolling allele. X with the recessive vision, dominant roller, X with the recessive vision, non-roller. Okay, now we can fill out this square and we can figure out how many of these couples' daughters would be expected to have normal vision and be able to roll their tongue, or how many of the sons would be colorblind and be able to roll their tongue. All right, we could make all of these kinds of predictions. Now, I'm not going to fill in this square right now because I don't want to make the video so long. The next one is going to be specifically about Punnett square practice. But see if you can fill this out on your own and then check in the next video for if you did it correctly. All right, so the last idea I want to talk about with respect to sex-linked traits deals with this idea of X inactivation. So in mammals, biological males are XY, biological females are XX. Well, females have twice the number of X chromosomes as men. Does that mean that all of their cells make twice as many of the gene products on the X chromosome? And the answer is no. So early in embryonic development for mammalian females, they undergo a process called X inactivation, where randomly in every embryonic cell, one of the X chromosomes is inactivated. And essentially, that inactivation causes that chromosome to condense into an inactive mass of DNA. Okay? Now, this is kind of interesting in tortoiseshell cats. All right, so the allele that determines their fur color is carried on the X chromosome. And so if a female is heterozygous for orange fur and black fur, normally black fur is dominant, okay? So, or we would think of it that way if this was an autosomal trait. But on the sex-linked traits, one of these X chromosomes is going to be randomly inactivated in every embryonic cell early in development when there's not that many cells. And so as a result, every time, if this is an embryonic cell that had the orange chromosome inactivated, all of the daughter cells resulting from mitosis from this original embryonic cell would only express the black fur allele. All of the daughter cells from this one would only express the orange fur allele. And so you're going to get this mosaic color pattern in tortoiseshell female cats. You're only going to see this color pattern in female cats because they are the only ones that have two X chromosomes. If they are heterozygous for the orange and the black alleles, then they're going to have this pattern. Well, what does that mean for, say, human females? The example that your book talks about is that there's an allele that the product of that gene is important in developing sweat glands. And so for females who may be heterozygous for the normal allele and the recessive allele, that they may have patches of their skin that produce sweat and patches that don't. Well, that's pretty mild, right? Not a very big deal. But there is an active area of research in X inactivation, and that there may be some X-linked disorders that normally would only be fully expressed in men if they get the recessive allele. But for women who maybe are heterozygous and they have that mosaic expression because their one X chromosome was inactivated early in development, there can be some women who may express more minor or milder versions of some of those X-linked traits as a result of X inactivation. So again,